Okay, so in work package 3, so what I prepared for you today, I just want you to know what we would like to do, what we can do, and there will be names of people who you can contact. So I will not present all the people that are involved in the project, but this is not to like diminish them. I just thought that for the sake of this this morning, it's much more important for you to know who you should address your questions, for example, for collaborations, than to know everybody who is involved. So as I see the VP3 or what we've put together, so in this work package, we want to concentrate on the host, on the microbiome, and on the diet. And for the host, what we have like in place, what we can use, it's the mouse, it's the piglet, and it's also the rhizus. Fungi, okay, yeah, it's a fungi, and yeah, we have a little bit of fungi and the roots there. What we definitely can do, so here you can see the battery of things we can do to these animals. So we can have them germ-free. This is the very top one. Or we can have them conventional SPF or even feral, thanks to the collaboration with the Charles University. And then definitely what we can do, we can just put one bacteria into the system, or we can do a defined consortium into the system. So this is the tool that we can provide. And then definitely we can control the diet. And here is the picture. So we can control each of these things to actually address the questions, how bacteria affect the host, how the host affects the bacterial communities, how the communities behave within the host, and we can push into that with the diet and see what happens. So we have two institutes involved in the VP3. It's the Institute of Institute of Microbiology and the Charles University, the Faculty of Natural Sciences. There is this laboratory of gnotobiogy where I come from. You can see it's very detached. Okay, so we are not based here in Prague. We are based in Eagle Mountains. Then there is the Laboratory of Cellular and Molecular Immunology, Laboratory of Fungal Biology. And in Charles University, we have Laboratory of Microbial Immunology and Division of Animal Evolutionary Biology. And so now I will introduce you to the people from this lab who you can contact or what they will do. So in Laboratory of Gnotobiology, this is the gnotobiotic thing, and you can see it's me, my colleague Dasha, Tomáš Hudcovic, and Marek. And what we do, we can do the germ-free and gnotobiotic piglets model. If you're interested in this, you contact Marek. We can develop with you the gnotobiotic protocols. We are really good at that. You can ask people who are already collaborating with us. Your primary contact for this will be Dagmar Šrutková or me. And what we can also do, we can breed for you germ-free and gnotobiotic mice. We can deliver them to you, and we can also provide the rederivation to germ-free conditions of your favorite genetic mouse background model. This will be, Tomáš Hudcovic will be your contact for this. If you forget these three, you can always contact me. I will redirect you. It's no problem. Here in Prague, there is Laboratory of Cellular and Molecular Immunology. Your primary contact will be Mila Kverka. Although he's not very much involved, he's the head of the lab. Or you can contact Klára Klimešová, Radka Roubalová, or Zuzka Zákostelská. And what they can do the microbiota analysis, animal models of human diseases. There is this new animal facility being set up. So I'm pretty sure that within 4 years, they will be able to do also some gnotobiotic things. Immune system profiling in vivo and in vitro, which means the cell culture system, different models of human diseases in mouse model, and also mouse to human translation. So they are good at tracking the biomarkers. And then there is the laboratory of fungal biology. So your primary contact here is okay, yeah, there are some people appearing there. Okay, so yeah, so it's Honza Jansa for this, and okay, some more people appearing, and also Caroline and yes, some Daniel. Okay, and what they will be interested in, it will be the gnotobiotic arbuscular mycorrhiza hyphosphere. Yes, yeah, okay, so this is what they will be interested in, and the major thing for me, or what sounds familiar to me, is the gnotobiotic. So they will really have a clean system with the ability to put bacteria inside. So from Charles University, we have collaboration with Laboratory of Microbial Immunology. The lead contact for you here is Jan Dobeš, although in this first year, he's not involved in the project due to many other duties he has. So actually his duties go to Dasha Šrutkova. So if you're interested in collaboration with Honza Dobeš, contact either of them. The thing is that for now, the established collaboration is that actually we are preparing for them the gnotobiotic and germ-free mouse models colonized either with defined bacteria or Candida albicans. And Honza is very much interested in the mucosal immune system development. And then we have the division of animal evolutionary biology. So your lead contact here is Jakub Kreisinger. Yes. Then there are also the other people from his team. And what they can do for you is the statistical and bioinformatic analysis of sequencing data, preparation of sequencing libraries, and other wet lab procedures, also establishing these. And from the running projects they have, so they are interested in evolution of murine microbiota and consequences of divergence of the microbiota between the laboratory and free living mice. So this will be the link for the feral mouse communities. So these are the projects, what they do, and the main contacts. And I also took the opportunity, because Honza suggested that we can talk about challenges. So what we have identified as immediate challenges. So first is the frozen opiac money. We cannot work because we cannot order. Second one is the tenders, which actually they are going for the machines, but then there are the tenders, for example, for the people. This is also with the unpredictable issues with the recruitment. For what they have in Prague here, so it's Mila Kverka's lab, so it's setting up the animal models in a new facility, which is ongoing. And for me to be able to collaborate with the other VPs, it would be good if we can just discuss how to communicate with other VPs, like what can be done. For the long term, so for a laboratory of gnotobioology, for us, it's definitely the location. As we are not based in Prague, so everything is harder. Then it's the bioinformatics. This comes from Mila, and from Mila also comes the unstable finances and paperwork overkill. So I think he was speaking just his heart out. Okay, and then I will give the microphone to Honza Jansa.